So welcome to video 2 in our course on RNA-seq and transcriptomics. And here we're going to look at mapping at splice junctions. So recall that a splice junction is the sequences that are define the end and start of a pair of exons that are adjacent to each other, where there's an intervening intron sequence. So what we're mainly talking about here is bulk RNA-seq in this course, where you have a bundle of different RNAs from which we prepare a library. We generate different reads that are in the form of cDNA. These are sequenced, and then they can be aligned to a genome as shown up there. This is our reference genome with the green blobs indicating different genes or exons. We map our reads to there, and we can look at the differential abundance of these different exons. And then we can consider that in terms of the counts that are present per isoform, normalized and all that. Or we can also look at alignment-free transcript quantification as well, where we have also information on the relative expression level of the different transcripts as well, without actually going through a remapping step. So mapping of splice junctions is key to all this. We have to consider that we have these different reads which are shown in black here. Typically they're paired end, and the reads lengths might be 100, 150 base pairs now. And then they might have some insert region in the dotted line that's might be 200 to 400 bases, give or take. And when we map the reads to different exons, if they're, say one read is mapping to this blue exon here, another read maps to this orange-brown exon, then we can kind of estimate the distance apart based on the insert size. That kind of gives us an expectation of how far apart they should be, and therefore which isoform they come from. So this is an example where you have 4 exons here and 3 distinct transcripts present. So you have the red-green transcript, the blue one, and then the blue kind of dark yellow one. So we can see that the left read of the rightmost pair, that's this one here, is mapping over this boundary. So it's mapping over the splice junction, and therefore we need to consider the type of splice junction that's present here at this boundary when mapping it to a reference transcriptome. So this is a reference transcriptome. It has the introns above it excised, but you consider the insert sizes now will be quite different compared to those above. And we consider for this read here that as mapping over this boundary, how it might map. So direct alignment to transcriptome, we'll see that there's no such boundary present, and it should map perfectly across this junction with no additional gaps. So there's 2 slightly different approaches in essence here, mapping to a reference genome and to reference transcriptome. So we know that some mappers that typically do DNA reads map to genomes, they can't see indent boundaries, but a splice-aware read mapper can consider these boundaries here between blue and green, and so on. So we can see that here, where we have 2 different reads, where we have these gaps that are allowed between the different reads in terms of the exons, and this might span one single read here in yellow and red, and that is a spliced alignment. That means we can consider all possible variants there that are kind of spanning different exons, and therefore infer the isoforms that are present from the collection of exons that are expressed. So Top Hat was one of the older programs that was first to do this. It mapped reads. They were initially classed as unmappable IUM reads at times, and these reads are mapped to putative exons, including the canonical splice sites as given here. So as part of that steps, it would join these exons that were separate gaps that were too small to be introns. Generally, introns are more than 70 base pairs apart, 70 base pairs in length, should I say. And then they were able to identify splice junctions from the map reads where there was splice sites between islands of different reads that were at least 70 base pairs or much longer apart as well. These islands were extended by 40 base pairs to allow for missed exon ends. And then these IUM, the unmappable reads, could be mapped to the ends of these exons to infer its splice junctions. So in this diagram here, what we're kind of seeing is that we have first read mapping of our RNA reads to a reference genome. We collect the unmappable reads, take the consensus regions by an older tool called Mac in this example. Next, the sequencing flanking the potential donor receptor splice sites could be found, and they form potential splice junctions. And these unmappable reads could then be mapped back to these junctions to try to see if they could be extended where possible. So those are all kind of central ideas that are now applied in more modern tools to look at splice sites associated with RNA-seq data. So please see the next video on RNA-seq experimental design.